We've talked about the sodium-potassium pump a little bit in some uses of ATP, but I want to delve a little bit more deeply into ATP and some of the pumps. So let's get into this. In review, the sodium-potassium pump is pumping sodium and potassium against their gradients, or you can even refer to it as uphill. But most importantly to remember is that they are moving against their gradients. That is to say, sodium that has a low concentration inside the cell is being pumped outside of the cell where there's a high concentration of sodium. In a similar fashion, potassium is pumped against its gradient into the cell. Keep in mind, potassium always, or nearly always, or should have a much higher concentration within body cells than outside of body cells. There's a couple exceptions to that rule, but by and large, we can consider cells having a super large concentration of potassium intracellularly and a much lower concentration extracellularly. But I think it's much more valuable to refer to it as the sodium-potassium ATPase, like we see right here. And the reason I think that's important is because the suffix ASE is informing us that this protein right here, this transporter, is an enzyme. And it's an enzyme that breaks down ATP, and we'll talk about it in a little bit. It actually makes ATP as well. So please keep in mind, in addition to these being transporters, and when I say these, the sodium-potassium pumps or sodium-potassium ATPase, they are an enzyme. That is to say, ATP is going to be split, or what is known as hydrolysis. ATP hydrolysis breaks ATP down into one molecule of ADP, that's adenosine diphosphate. It was originally adenosine triphosphate, tri meaning 3, adenosine diphosphate meaning 2 phosphates. ATP is hydrolyzed or broken down into ADP in this one inorganic phosphate. We refer to it as inorganic because it is an inorganic element. So ATP is broken down into ADP in that one inorganic phosphate. That in and of itself doesn't really do anything for us or do anything for the cells unless we mention and stress that the breaking of this bond is going to release energy. As we've talked about, these 3 phosphates, 1, 2, and there's the third one down here, these 3 phosphates are all negatively charged. They don't want to be around each other. The molecule is somewhat like a loaded spring. That is to say, it has all this pent-up energy. And if a bond is broken between the phosphates, a bunch of energy is released, and that's what we see right here, and it's liberating that one phosphate. So this actually, with the broken bond, is the ADP, and this is that inorganic phosphate. What are the roles of the sodium-potassium pump? Well, once again, it's to maintain chemical gradients. That is to say, a higher concentration of sodium outside of cells and a higher concentration of potassium inside of cells. We need this for metabolic activity, for the cell to change voltage or to depolarize. And that's going to happen constantly, all day long, all minute long, all hour long. In the time I've been talking here, it's probably happened thousands of times in all of us. So as sodium comes in and potassium leaves, that's really good for depolarizing cells and repolarizing cells. But we need to restore those gradients, and that is achieved by the sodium-potassium ATPase. Another role of the sodium-potassium ATPase is to maintain the electrical differential of the cell, or the voltage. That is to say, cells have a positive charge on the outside of the cell and negative charge on the inside of the cell. Okay, now that I have that drawn in here, positive on the outside, negative on the inside, that is a voltage or difference in charge between the outside and the inside of the cell, inside of a resting cell. So we want this electrical differential to be present in resting cells. And the sodium-potassium ATPase helps to maintain that. Now, how does it help to maintain that? Well, because it's pumping more positive charge in the form of sodium. It's pumping more positive charge outside than is being pumped inside. We're only pumping 2 positive charge in with potassium and 3 positive charge out with sodium, meaning the outside of the cell is going to be more positive and the inside of the cell is going to be slightly negative or at least less positive. Now, one thing I do want to stress is that I'm saying 3:2. That is the ratio for the sodium-potassium ATPase, otherwise known as the sodium-potassium pump. But I want to be clear, there's not just one sodium-potassium pump in every cell. There's probably hundreds, dare I say thousands. So there's a bunch of sodium being pushed out and a bunch of potassium being brought in. This is active transport. It utilizes ATP, which is a form of kinetic energy. Within the human body, we generally use just 2 forms of energy, chemical energy and kinetic energy, which we'll talk about in a bit. The main form of chemical energy we utilize in the body is via ATP, specifically the hydrolysis of ATP. And a third role of the sodium-potassium ATPase is to maintain cell volume. In addition to having ions within cells, there's a number of protein anions, that is to say negatively charged proteins in the cell, in addition to phosphate ions. These protein anions and phosphate anions help contribute to the negativity within cells. But please understand that these proteins and the phosphate, in addition to other roles they may have, are functioning as solutes. That is to say, anything suspended within the cell, within the fluid, within the intracellular fluid, is a solute. Sodium's a solute, potassium's a solute, and these are solutes, the proteins and the phosphate. Now, as far as cell volume goes, and this is related to cell volume, if the sodium-potassium pump were 3 for 3, that is to say pumping 3 sodium out and 3 potassium in, we're going to have more solutes within this cell, including these protein anions and the phosphate ions. And we don't want that because then water would be drawn into the cell. And as water passes the plasma membrane and continues to move into body cells, it causes those cells to swell, and in worst case scenario, they're going to burst or lyse. So by pumping 3 sodium out and only 2 potassium in, it's ensuring that there's equal concentration of solutes on the outside versus the inside of the cell. If, under certain conditions, and these do happen independent of the sodium-potassium pump, when conditions of the cell starts to swell because water is moving into the cell, it actually activates the sodium-potassium pump to pump more sodium out of the cell than potassium in. And by continually pumping sodium out of the cell, we will get water chasing the sodium out of the cell. Water tends to chase solutes, so this will help maintain the cell volume. Specifically, it will prevent the cell from swelling with H2O. Now, the really cool and interesting thing about the sodium-potassium pump is that it will work in reverse. That is to say, at rest, when there's ample amounts of sodium outside the cell and a little bit of sodium inside the cell, and the converse with potassium, as sodium's moving into cells and potassium is moving out of cells, they create the energy of motion, which is kinetic energy. Kinetic energy is produced when things, ions or molecules, are moving down their gradients. And as sodium's moving down its gradient into the cell and potassium moving down its gradient out of the cell, that is not these arrows, by the way. These arrows are the sodium-potassium pump working in the opposite direction. When sodium and potassium are moving down their gradients, they create the kinetic energy that is going to be utilized by the sodium-potassium ATPase to make more ATP molecules. And that process is known as condensation. Condensation is the opposite of hydrolysis. Condensation of ATP takes one ADP molecule, coupling it to an inorganic phosphate in the presence of energy, and in this case, this is the kinetic energy created by sodium or potassium moving down their gradients. That energy is used to make more ATP. So the really cool thing about the sodium-potassium pump is it works in reverse. It breaks down ATP and it also makes ATP. So let's take a look at some more examples of ATP functioning as a pump and delve more deeply into the kinetic energy and secondary active transport. Another example of a pump utilizing ATP to move things against their gradients is the calcium pump. And once again, it's also referred to as the calcium ATPase. This is representing a muscle cell. This is an organelle known as the sarcoplasmic reticulum that stores calcium. Now, this example is a little bit different because the sarcoplasmic reticulum is an organelle within the cell. It is intracellular. It stores calcium, and to facilitate muscle contraction, calcium is required to leave the sarcoplasmic reticulum and move into the cytosol, cytoplasm, or the intracellular space. But that calcium needs to be returned to the sarcoplasmic reticulum, and that's achieved by the calcium ATPase, which is moving via the chemical energy of ATP, moving calcium against its gradients into the sarcoplasmic reticulum via the calcium pump. Another example is what happens in the stomach. So if we look over here, this is a very basic drawing of the stomach. And you may remember that we've talked about the pH of the stomach being very low. It's roughly around 2. If you were to pour stomach juices, otherwise known as gastric juices, on your skin, it would burn your skin. So there's a low pH within the lumen or the open space within the stomach. And that low pH means there's a high concentration of the hydrogen ion. And that's what we want for immunity and digestion. The hydrogen ions are secreted, and that helps break down proteins during digestion. Now, those hydrogen ions, also known as protons, keep in mind, a hydrogen ion, it is often referred to as a proton. So this proton is moved or pumped into the lumen, once again, for immunity purposes and for digestion. But because there's such a low pH here, that means the hydrogen ion is moving against its gradients, and that's what I'm showing right here. This is a parietal cell. These cells lining the stomach are parietal cells. Not all of them are parietal cells, but this example is of a parietal cell. And the movement of hydrogen ions into the gastric lumen is achieved by the hydrogen-potassium pump, otherwise known as the hydrogen-potassium ATPase, which is pumping hydrogen out of the cell and potassium into the cell. But hydrogen is being pumped out because there's such a low pH in this area. And this is what's known as a proton pump. Individuals who take proton pump inhibitors are blocking the activity of the hydrogen-potassium ATPase, otherwise known as the proton pump. Individuals take proton pump inhibitors if they have gastric problems with a lot of acidity. So this is another great example of the use of ATP to move things against their gradients. Like I said, we utilize kinetic energy as well to pump things against their gradients. Kinetic energy is the energy of motion, and anything, ions or molecules, moving passively down their gradients create kinetic energy. In this image right here, this is a cell within the kidney, specifically within a structure known as the nephron. And within this cell right here, like most cells in the body, there's a high concentration of glucose within the cell and a low concentration of glucose outside of cells. And as normal, low concentration of sodium inside of cells, high concentration of sodium outside of cells. So without getting into the details of why, please understand that the cells of the nephron need to move glucose into these cells. Because it's moving against its gradients, some form of energy is going to be needed, and the body, these cells utilize kinetic energy. That is to say, sodium moving down its gradients from an area of high concentration to an area of low concentration is going to create this kinetic energy to pump glucose against its gradient. This transporter is known as the SGLT, which stands for sodium glucose transporter, that utilizes kinetic energy to pump glucose into the cell against its gradient. Now, one thing to keep in mind, as I've described this, no ATP is needed for this process. The SGLT is utilizing kinetic energy. But it's very important to understand that if we didn't have the sodium-potassium pump functioning here, I realize I don't have the sodium-potassium pump drawn in this image right here, but please keep in mind, nearly all cells, dare I say all cells of the body, have sodium-potassium pumps to maintain their chemical gradients. So the sodium-potassium pump is working in this cell as well, the cell of the nephron. And by doing so, it's maintaining sodium's gradient so it can then produce that kinetic energy as it's moving into the cell. If this cell did not have a sodium-potassium pump utilizing the chemical energy of ATP, then we would not be able to have sodium passively moving into the cell. When cells utilize kinetic energy to move things against their gradients, this is known as secondary active transport because it's not directly using ATP. But please keep in mind the primary active transport using the chemical energy of ATP needs to be happening as well. We can also describe the sodium glucose transporter as a symporter because it's moving these two entities in the same direction. We also have antiporters right here. This is known as the sodium-calcium exchanger, and it's known as an antiporter because it's moving entities in opposite directions. In this example, sodium into the cell down its gradient, and calcium out of the cell against its gradient. We find the sodium-calcium exchanger within myocardial contractile cells. These are the muscle cells of the heart that help the heart contract. And the NCX is utilizing the kinetic energy created of sodium moving down its gradient to pump calcium against its gradient out of the cell. So once again, kinetic energy is utilized for secondary active transport. Chemical energy is utilized for primary active transport. We can't really have kinetic energy without primary active transport or chemical energy first establishing those gradients.